Okay, why don't we get started now? So good morning, everyone. I'm Paul Danis with the consortium, and we're pleased to have Fabio Gomes with us today. Let me just give you a couple little notes of introduction, and then we'll get going with the seminar. First, we want to thank our sponsors that enable us to have these kinds of programs, Thermo Fisher, Brooker, SciEx, and Lilly, and generously our major sponsors, as well as support from Agilent and Pfizer. So we thank them. We thank some of these people, maybe you're online, some of the affiliates of these organizations. So we thank you directly. And if you see these company people around somewhere, just send them a thanks and say, we appreciate your help for allowing us to do these kinds of things. But just a couple of bits of news. Some of you may have seen that we recently published the paper about our initiative on top-down of native proteoforms and complexes. This paper came out in Nature Methods. It's actually available online. You can take a look at it if you don't have a subscription. If you go to our website, you'll find links there where you can read the paper. And it's meant really as a on-ramp. It's sort of two. One is like kind of assessing the state of the art, like what's going on in native today, like what the machines tell you and what the workflows tell you. And the other is to give some basic protocols and approaches for people that want to either optimize what they're doing or else even start native work. We know it's a little challenging. So there's information there on the workflow details, a basic workflow to get you going based on what kind of information you're looking for, some representative data from the different instruments, the kind of coverage you can get from different instruments on different types of molecules, different sizes, as well as some real details around tuning parameters, like your way to start tuning for these things. So for a bunch of the different instruments there, there's real details about how you can set your instruments up if you want to do this kind of work. So you're welcome to go take a look at that paper. As I mentioned, that's on our website. There's a link to it there. And next month, we'll have Novera Alam from Northeastern. She'll be speaking about some of her recent work. And I'll have details on that out to you folks in the near future. And if you're interested in presenting some of your work, if you have some nice work you think the community would love to hear about, either you can put a comment in the chat and say, hey, I'd like to talk to you about stuff, or you could just send an email to us at the consortium or to anyone on the board or like that. Let us know, and we'd be happy to just to have you involved with this. So without further ado, let me turn it over to Dr. Fabio Gomes. He's currently in the Department of Chemistry at the Virginia Commonwealth University, and he'll be speaking to us today about some of his work on proteoforms and complexes in cancer cells. So go ahead, Fabio, it's all yours. And as far as questions, you can either you can ask them live after he speaks, or you can throw a comment in the chat if you'd like, and we'll address it there. So go ahead, Fabio, it's all yours.
Okay, thank you very much, Paul, for the nice introduction. So can you all see my slides? Can you see my slides?
Not yet.
Not yet.
So I have to share again.
Yeah, it looks good. Thank you.
Okay, thanks for the introduction, Paul. I'm so excited to be here to give a talk for this community. So I will initially talk a little bit of what I do here in my lab, what I intend to do. So I develop analytical platforms that combine chromatography, capillary electrophoresis, ion mobility, and mass spectrometry to probe intact proteoforms and their complexes within cells. So the goal is to apply these platforms to better understand the molecular mechanisms that govern ligand efficacy, drug resistance, and how antagonists bind and modulate the biological activities of important drug targets, such as membrane proteins. So we hope that with the acquired knowledge, structural knowledge, is then help us to identify therapeutic targets, advance the drug discovery field, and ultimately improve public health. So these are the technologies that I have in my lab. I have a chromatographic system that was placed as alone by Agilent. This chromatographic system allows us to do size exclusion chromatography. It has a mouse detector and also a GAD detector, a diode array detector. So there is also a company that is called CMP Scientific that placed a capillary electrophoresis in my lab. So this is a very versatile technology that we can use for native separations and denatured separations as well of protein assemblies. So we have a nanoliquid chromatograph, and we also have a QE UHMR and a Synapse G2SI that I inherited. And then I upgraded this system for large molecules. Oops. So most cellular functions, before going to the native top-down strategy that I developed while I was a postdoc at the EATS lab, I will talk a little bit about proteins. I know this community is very familiar with proteoforms, and they're complex, so I won't be extending. It's just a short introduction. So most cellular functions are driven by protein complexes that are often assembled via non-covalent interactions of monomeric protein subunits. These monomeric protein subunits can undergo two numeral structural changes that will allow numerous proteoforms. So proteoforms basically is distinct protein forms from a single gene product. So, and it is estimated that in the human organism, it's over 1 million proteoforms. So this is due to post-translational modifications, mutations, truncations. So imagine how the complexity that this can generate in the cellular environment, as there are so many post-translational modifications. So this paper says over 1 million, but I believe is much more than 1 million proteoforms. And these proteoforms can also modulate biological functions independently. For instance, the tau phosphorylation is well known. Hyperphosphorylations are well known in Alzheimer's disease. So the GIG group from Wisconsin has consistently shown the power of proteoforms as a potential biomarkers in cardiac disease. For instance, the troponin protein is hyperphosphorylated, undergoes two numerous phosphorylations. So, and these proteoforms, they can interact to each other and form complexoforms. Complexoforms I will call in this talk as different proteoforms that form protein assemblies. So in this example here, you have three different proteoforms, but you can have a dimeric assembly, you can have a trimeric assembly, and so on. So how we measure these proteoforms? So top-down proteomics is the most common method to interrogate these structures, and it is widely accepted the advantages of top-down over bottom-up proteomics. So with top-down proteomics, you can interrogate the primary structure of the protein. And the beauty of this strategy, this mass spectrometry-based strategy, is that you can see the entire picture of the protein. So, but this technology is usually used to measure the primary structure of the proteins or the proteoforms. So another technique, another mass spectrometry-based strategy is called native mass spectrometry. This technique is used to measure intact protein assemblies. So with this technique, it is possible to observe protein-protein interaction, protein cofactor interactions. You can see the stoichiometry of the complex. So, and this is usually, you measure protein assemblies via non-covalent interactions. So these two techniques has been combined. There are two different ways to do it. So one way is Kelleher group disassemble protein assemblies into monomeric subunits. The other way, it is Lu group who fragmented the entire macromolecular assembly. So, and since then, like there have been a few papers where they interrogated protein assemblies in samples with biological relevance. For instance, Kelleher group developed a native top-down strategy to interrogate endogenous proteoforms from complexes. The some group developed a capillary electrophoresis strategy, and recently the G group developed a strategy to interrogate troponin complexes from human heart. So, but this strategy is still very difficult, and I think there are few things that impede its widespread acceptance. For instance, these complexoforms, proteoforms in general, and complexoforms, they are found at very, very low concentrations in the cellular environment. The separation of these compounds are very difficult in complex mixtures like cells, and fragmentation of this is still a problem. Bioinformatics is not really still, there is more room for improvements. Although there are some software, like we're gonna talk a little bit more, ProCyte native, there has been, I used this software, and there is another software from Wisconsin, which is called Mesh, but I will limit this talk for the use of ProCyte native, which is the software that I used in this research. So the goal of this research, as mentioned previously, I started this work when I was a postdoc at the Yates lab. It has been a very, very long time cooking. So the paper is under review in Nature Chemical Biology. So I think we are about to finish some of the last experiments, and then maybe the paper will be eventually published. So two reviewers are satisfied, but one is still asking for the biological experiments. So the goal of this work was to develop a native top-down proteomic strategy to characterize endogenous complexoforms in MC F7 cells with any thought overexpression of epidermal growth factor receptor, which is a model of resistance, characterize endogenous complexoforms that are directly associated with development of drug resistance, and also correlate the proteoform diversity with specific breast cancer cell physiology. So how we did this? As mentioned previously, there are different ways to do it. The way we did this was in the first stage using multiple rounds of mass spectrometry. For instance, in the first round, we detect the complex, the protein assembly. In the second round of in the MS2, we disassemble this complex using gentle collisional energy. And in the third stage of the mass spectrometer, we fragment the monomeric protein subunit. So this is the workflow of the strategy. So how I lyse the cells? I used a Downsing homogenizer. In this Downsing homogenizer, I used a buffer which is under near native conditions. So after lysing the cells with Downsing homogenizer, the cell lyser was injected into the AGPLC for fraction collection. So after fraction collection, each fraction was concentrated using kilodalton filters and then injected via infusion. We used a chromatographic system to infuse the samples. So there was no chromatography, we just used the system to infuse the samples using a capillary, and we were able to infuse proteins for more than an hour. So for each infusion. So we then used Orbitrap fusion lumos. We extensively optimized this instrument to accommodate protein complexes up to 70 kilodaltons. And then we used finally we used the ProCyte native to analyze the data and identify proteoforms of the complexes. So we compared, as mentioned previously, two different breast cancer cells, one with overexpression of EGFR. So our strategy is based on we developed a strategy. Actually, the method native size exclusion has been developed by YSOC lab, and the GI lab also concentrated samples from the nature proteoforms. So what I did was combined these two strategies and developed my size exclusion chromatography strategy. So with this strategy, I was able to concentrate the protein complex. So I first developed a strategy using three protein standards that ranged from 6 to 17 kilodaltons. And then I injected each sample 17 times. So the high reproducibility of the system allowed us to concentrate the protein complexes. So we after that we collected each fraction and run a native gel to confirm if the proteins were if we still have non-covalent interactions of protein assemblies after size exclusion. And as you can see in the two gels, proteins are still under near native conditions. So, and this represents how we collect each individual fraction. So using this strategy, we were able to identify about 100 complexoforms from 17 protein assemblies using our nano famous MSN strategy. So we identified the endogenous 3-thiophosphate isomerase, which is a dimer. This dimer is about 50 kilodaltons, as you can see here. If you look at the MS1, we were able to identify the dimer with 2 deamidations in each of the monomeric subunits. So, and if you increase this charge state here, you can see that there is a lot of things going on. in this charge state. So we were able to identify truncated complexes of this of trioxephosphate isomerase, and we identified the complex with one phosphorylation and the other complex with two phosphorylation. So we further selected the charge state 15. We disassembled the complex and we fragmented the charge state 11 in the MS2. So we obtained this fragmentation map. As you can see here, we were able to identify two deamidations. So these two deamidations was identified in each of the monomeric subunits. So it has been reported before, and top-down confirms that this protein is deamidated. As you can see, we were able to obtain strong fragmentation that flank each side of the modification. So next, we also interrogated a trimeric assembly, so which is called the migration inhibitory factor. So this is a trimeric about 37 kilodaltons. So as you can see here in the MSU1, you can see several different complexoforms. We disassemble this trimeric structure, as you can see in the MS2, and we were able to characterize this, the monomeric protein subunits, and they are combined in several different ways. So we identified the modified form. We also identified a nitrosylated form, and we also identified acetylation that form complexes in several different ways. So next, we characterized a protein metal, which is called the superoxide dismutase, which is about 32 kilodaltons. As you can see in the MS1, we can see, we aimed to see if we can characterize protein metal interactions. So when we disassembled the complex, the dimeric structure, we were able to see the metal is still bound to the protein. So if you look at the fragmentation maps, so you see the Apple sequence where it has acetylation in the N-terminus, but without the metal. So if you add the metals, which will account to the hollow fragment ions, you can see that the sequence changes the pattern. So we confirmed this using the TD validator software. We cannot really say the location of these these metals, but we can basically say that this is the region that they are found, and also this is consistent with the literature. Due to the lack of strong fragmentation, we cannot really characterize the individual position of each metal, but again, this is consistent with the literature. So next, we characterized the nuclear transport factor, which is a dimeric structure. We were particularly interested in this structure because these proteins transport proteins from cytoplasm to the nucleus, and it is related to, we decided to see if this affect the estrogen receptor alpha, which is a nuclear receptor. So we did several biological validations to confirm our observations, but as you can see here, the complex, there are several proteoforms that form different NUF2 assemblies. And if you look at the MS1, you can see the black, which is the structure that was found in MCF7 cells, and the The other color is the one that was found in EGFR. So you can see clearly the EGFR affect the dimerization of nuclear transport factor 2. And when we interrogated the MSC2 structures, were clear, like when we disassemble the complex, it is clearly observed the dimerization is affected. There is a very clear difference between the proteoforms from MCF7 and the proteoforms from MCF7 EGFR. So, and these proteoforms showed demonstrated based on our biological experiment to affect the activity of the estrogen receptor. So we next characterized different NuFi2 interactions with SOG1 and several other proteoforms. So there are so many things going on in the cellular environment. So NuFi2 interacts with other proteins. So the key finds was this top-down, native top-down platform opens new avenues for large-scale native top-down characterization of proteoforms and their complexes, and it is uniquely positioned to, one, provide the identity and connectivity of intracellular proteoforms in protein assemblies, to illuminate differences in expression patterns of proteoforms and their complexoforms, to define the molecular basis of the structure-function relationship of proteoforms and then complexoforms. So epidermal growth factor destabilizes nuclear transport factor 2 dimers, which inhibits the NuFi2 activity and contributes to EGFR driving tamoxifen resistance. The K4 and K55 post-translational sites discovered with differentially impact the effects of NuFi2 and inhibition of the estrogen receptor signally, the characterization of endogenous proteoform interactions revealed the molecular diversity of complexoforms. So advancing the understanding of the higher order organization of the cells provides rational drug design to overcome drug resistance in breast cancer. So what we are doing now, in my lab, we are interested in breast cancer. We are particularly interested in estrogen receptor alpha, which is the major target for breast cancer drugs. So we believe that drug resistance occurs when groups of cancer cells with a tumor contain molecular changes or proteoforms that make intensive endocrine treatments. So we are currently estrogen receptor, once activated, it dimerizes. So there are several proteoforms of the estrogen receptor, include the truncated forms of this receptor. So interesting here is that tamoxifen, it is antagonist for the full-length estrogen receptor 66 and agonist for estrogen receptor alpha 36. So as you can see, there are many post-translational modifications, and it can be identified in different subcellular locations. So we hypothesized the biological actions of estrogen and anti-estrogen drugs in development of drug resistance are regulated by estrogen receptor alpha, their complexoforms. So towards this goal, we want to characterize how estrogen receptor proteoforms are regulated by different ligands. We want to define these macromolecules in normal and resistant breast cancer cells. We want to identify these molecules, interrogate these molecules in different cellular locations. So we are currently optimized optimizing our conditions. So Kanshang, my graduate student, she's working very hard. We are initially developing SOPs using size exclusion, using reversed phase chromatography, using native capillary electrophoresis, and native top-down mass spectrometry based strategies. So I would like to thank my research group, VCU Startup Funding, EH Lab, the Netless Lab, which was the lab that was doing the biological experiments, the Protein Assays, which is the company that provided the software tools, Agilent, which is the company that placed LC in my lab and will allow us to interrogate several different complexes, and CMP Scientific and Thermal as well. And thank you all for your attention.
Great, thank you, Fabio. Interesting stuff. I just had a quick question on your workflow. It's nice you're using all commercial tools,
Yes.
combined in a certain workflow. So it's not like someone has to invent something. So if someone wants to employ this kind of workflow, what do you think are the biggest challenges in implementing this, since that's something you've done?
So the workflow is quite simple. I mean, it's very difficult, like to get these things working, but the workflow is not something really complex, because the idea is to make simple and reproducible so other labs can easily reproduce. But as long as you have the right tools, for instance, in this case, we optimized several conditions for the Orbitrap fusion lumens. But one limitation of this work is that we were limited to proteins up to 70 kilodaltons. So if you are interested in small proteins, protein assemblies would be a good strategy, this workflow, I think.
Okay, thanks. Okay, I have a question from Joe Liu. It says, nice project. Fabio, the EGF overexpressed cell line reduced the abundance of the NUF2 dimer complex. From bottom-up proteomics, is the level of NUF2 also lower in the EGF overexpression cells?
So we observed, actually, one of the reviewers asked to show different methods, and the NUF2 EGFR, yes, indeed. So the other techniques were, we achieved the same results with using the other techniques. Yeah, including the biological experiments that we did. We observed the NUF2, the levels of NUF2 was decreased by EGFR overexpression.
Thanks. Another question from Amin. Great talk, Fabio. Have you ever tried to get intact EGFR only from breast tissue homogenates? Another question, have you used any detergent for this project you just presented?
So I think this is very interesting, and this is definitely something that I want to target, but this is a very difficult target, and I definitely have this in mind. This is a membrane protein, which is 180 kilodaltons, about 180 kilodaltons. Yes, I definitely would like to target this sometime in my lab. Because when we developed this work, the instrument, like this Fusion Lumos, after several optimizations, I could not see anything bigger than 70 kilodaltons, 70.
Okay, now the question part about detergents.
Was that?
On the second part, have you used detergents in this project?
So because we look at soluble proteins. So the size exclusion strategy was based using ammonium acetate, 200 nanomolar, and we then when we transferred to the mass spec to run the samples in the mass spec, all we did was to concentrate the samples using kilodalton filters. So we did not use a detergent, but this is something that we are thinking now.
Great, thanks. Question from Yuzo. Great talk. May I know this platform's reproducibility? Do you have QC control? How about sensitivity, and what software are you using for native top-down data analysis?
So yeah, that's a great question. So we, like for the size exclusion strategy, we repeated, and the success of this strategy was based on one of the reasons was because of the high reproducibility for the size exclusion. But for the mass spec, we considered a minimum of, like, for instance, the complex to be identified in at least 2 samples. We also used p-score cutoff, this type of controls.
Okay. And then the question about the software for native top-down.
So we used this ProSite native, and we had some controls, as mentioned previously, like a p-score we used, and other controls, like for metals, for instance, like if we identified more than more than 5 or 6 metal bindings in the protein, we would not consider. But the major control we had was to develop a strategy before using protein standards. So we used carbonic anhydrase. We used the carbonic anhydrase, which is a protein about 30 kilodaltons and has a metal on this protein. We used avidin, which is a glycosylated protein, which is about 66 kilodaltons, and we used another protein that I forgot the name, but it's a tetramer. So avidin is a tetramer, 65 kilodaltons, and the other steptravidin, which is another tetramer and is about 50 kilodaltons.
Okay. I just, on the software, you said the ProSite native. I assume that's a
Yes.
thermo only.
Was that?
I assume that's for thermo instruments only.
Yes, it is.
And you had also mentioned about MASH. Have you looked at the MASH
So
software as well?
I've been looking into MASH now, but when I was working, this work actually was a very long time cooking. So they already published one paper using ProSite native. The company published one paper, I think, with the Robinson group, and this is only for thermal instruments, but it is not commercialized by Thermo.
Okay. All right. So Joe Lou has another one in there. He's asking the simple question, which has a complex answer. Is there a way to integrate bottom-up proteomics with top-down proteomics?
I think they are complementary to each other, but I think it is good to do both if you have the chance. But I think top-down, it's really clear. The picture that you see of the complexes of the proteoforms, but it is certainly a strategy to complement this workflow. So one of the reviewers, he asked for other techniques that complement this workflow, these observations that we saw with the mass spectrometer.
Great, thanks. I just had one other thing I was wondering. On your coverage maps, there seemed to be some had excellent coverage and some had much, much less coverage. What do you think the main factors were? And is it resistance to cleavage or some other things?
So some proteins, they obviously fragment better than the others. But this one advantage of this workflow is because I infused the samples and I could spend a lot of time, like fragment the samples, accumulating accumulating ions for a lot of time. That would not be possible if I have a LC, liquid chromatography, or like size exclusion, or capillary electrophoresis strategy. So this is one of the advantages why we got very good fragmentation for some complexes, but it's still very hard to fragment. And I think it is protein dependent. Like some proteins can break very easy, others and just very difficult.
Yeah, I see. Eric has a question.
Yes.
Eric, you can go ahead.
Sure, Fabio. Thanks. Great talk. I was really like that. And I have a couple of questions. One is regarding this slide. When you mentioned that you could only optimize the MS method or the MS conditions to detect 70 kilodalton proteins, it's not a problem of, just to double check, it's not a problem of fragmentation, right? So you couldn't ionize or detect in the mass spectra protein complexes beyond 70 kilodalton. Is that right? And if that's right, why was that? Or why
So
do
I
you
think it
think
is
that
just a
happened?
limitation of the instrument, like the Orbitrap Fusion Lumos. I was able to see after several optimizations, I was able to see 70 kilodaltons, but there are other instruments, like newer instruments from Thermo now, they did some hardware changes where you can see larger complexes, but I think fragmentation is still a problem. So there are some groups like Dr. Broadbelt from Texas that has a very power laser, UVPD laser. She has been successful fragmenting proteins, but I think it is more, even if you have an instrument that can allow you measure larger stuff. Actually, Joe Lu, Dr. Lu, he in this paper that he published in Nature Chemistry, I think it was 2017, he fragmented very, very large protein assemblies. And the beauty of his experiments is because he fragmented the entire complex.
Yeah, I think I read the paper. I think you're talking about beta GAP, if I'm correct. Is that the paper that you're talking about? Yes. And then the second question is, when you mentioned the truncated form of certain protein or protein complexes, when you extract that from tissue, how do you figure out or work out your way to determine that, okay, this is a truncated form? I mean, I can imagine how, but could you please walk me through?
So truncated, as you know, it is a protein that doesn't have a part of the sequence. So in my experiments, I use protein inhibitors, which can minimize the like artifacts. But actually, we can see the truncation. There are so many, like in biological samples, but this is a biological process. And to make sure, I use like ProCyte Lite, which is another bioinformatic tool. This is free. So you can add your MS and you can your MS, and you can check the sequence, the protein sequence. But it's very hard to characterize these truncated forms.
Okay, I see. All right, thank you.
No problem. Thank you.
Great, thanks. Another question coming from Amin. For TD peak assignment, have you confirmed each of the peaks? In other words, what about FDR? Says we have done TD annotation manually for carbonic anhydrase and some other proteins, and I've always seen that manual annotation has always had less false annotation compared to any commercial software.
So this work was a lot of manual interpretation to conform the spectrum. And as I told you, as I mentioned previously, we used some cutoffs like protein characterization scores and the fragmentation patterns of the protein, but we did not use FDR. So the software does not calculate FDR.
Great. Any other questions, comments for Fabio? Okay, if not, then thank you again, Fabio, for sharing your work. It's interesting work, challenging for sure. We wish you the best of that and hope to hear some great results in the future. So thank you again, everyone, for attending, and we'll have more information about the August seminar coming up shortly. Thank you all, and have a great day. Bye-bye.
Oh, thank you very much.
You're welcome.
Thank you, Paul. Have a good day.
Thank you, Paul.
Thank you, Fabio. Very nice.
Have a nice week.
Thanks, you too. Take care.
Bye. Take care. Bye.